The bacterium Agrobacterium tumefaciens is a natural genetic engineer of a wide variety of plants. The bacterium has a loop of DNA called a tumor-inducing or TI plasmid and can transfer part of this plasmid into a plant cell. The DNA transfer induces plant cells to make food molecules for the bacterium. Agrobacterium, which is ubiquitous in the soil, begins to communicate with plant cells at a wound on the plant body. The wounded plant releases phenolic compounds that, depending on the species involved, Agrobacterium recognizes. In the cell envelope, a protein called Vir-A detects the chemical signal from the wounded plant. The binding activates Vir-A, which can then activate another protein called Vir-G through phosphorylation. A number of genes called Vir genes are located on the TI plasmid. While the genes encoding Vir-A and Vir-G are turned on all the time, the expression of the other Vir genes requires activated Vir-G as a transcriptional activator. Two of the gene products are Vir-D1 and Vir-D2. The Vir-D proteins are endonucleases that cleave at the 25 base pair sequence repeats at the ends of the tDNA. As a result, the cell produces a single-stranded tDNA molecule attached to Vir-D2 at one end. Vir-D2 directs the transfer of the tDNA into a plant cell in a process similar to bacterial conjugation. Within the plant cell nucleus, the tDNA becomes integrated into the plant genome. A number of genes are encoded in the tDNA and become expressed in the plant cell. Some of the gene products are responsible for the synthesis of the plant hormones cytokinin and auxin, which trigger the plant cells to divide rapidly and form an undifferentiated mass known as a crown gall tumor. The tDNA also encodes genes that control the production of unusual amino acid derivatives called openes. In return for delivering the tDNA to the plant, the bacterium gets the openes as a source of carbon and nitrogen. Researchers employ Agrobacterium to genetically engineer a wide variety of plants. In this procedure, the tDNA is placed on another plasmid that is more easily manipulated, and the DNA between the flanking sequence repeat regions is replaced with a gene of interest. Upon infection, the virulent system induces transfer of the tDNA to the plant cell without any tumor-inducing genes, allowing genomic integration of the recombinant gene without tumor induction or opene production.
আপনাদের today we will start a new chapter, chapter 3. The name of chapter: Plant Genetic Transformation, Prospects and Potential. Different genetic material transfer by the different molecular mechanisms for the development of different types of traits, for the production of hybrid resistance plant, insect resistance plants, and for the production of different types of hybrid by the transformation of plant genetic materials. First, genetic transformation. What is genetic transformation? Genetic transformation is the process of integrating exogenous DNA into the germline of whole organism so that it is inherited in subsequent generations. Transformations of cell is a widely used and versatile tool in genetic engineering and is of critical importance in the development and molecular biology. The purpose of the technique to introduce a foreign plasmid into plants. The plants then amplifies the plasmid, making large quantities of it. So, due to the genetic transformation of foreign DNA with the insertion of plasmid DNA, we will make a large quantities of a specific DNA and it also evaluated by the expression of the transferred DNA. For the expression of the specific traits and the specific hybrid and also the specific properties of it. So genetic transformation is nothing, just transfer a specific gene or a specific DNA fragment as a tDNA and also in the different forms of DNA with the attachment of plasmid. It is genetic transformation in plants. Next, plant genetic transformation prospects and potential. How many significance and how many importances we found after the genetic transformation in plants? First, gene transfer or uptake of DNA refers to the process that makes a specific pieces of DNA into cell. It is widely used and versatile tool of genetic engineering and is critical importance in the development of molecular biology. Next, it is used to introduce a foreign plasmid into plants. The plant then amplifies the plasmids, making large quantities of it. Transgenic plants are plants that carry the stable integrated foreign DNA. It is used to make multiple copies of DNA called DNA cloning. Last, it is used to genetic alteration of the cell resulting from direct uptake or incorporation of the exogenous genetic material from its surroundings to the cell membrane. So, in the point, you read 4 or 5 easy points for your semester final exam. Next, plant transformation. What is plant transformation? Plant transformation is a way to insert DNA from another organism, normally another plant, into the genome of a plant of interest. Plant transformation is now a core research tool in plant biology and is a practical tool for transgenic plant development. Techniques for plant transformation. How many techniques used in the plant transformation or transformation of genetic materials in the plant? Plant transformation with TI plasmid on Agrobacterium tumefaciens. So first to use a specific DNA with the help of TI plasmid on Agrobacterium tumefaciens. TI plasmid, the name of full name TI, tumor inducing plasmid, that is generally found in Agrobacterium tumefaciens. TI plasmid derived vector system, physical methods of transferring gene to plants, chloroplast engineering, use of reporter genes in transform the plants, manipulation of gene expression in plants, production of Manhattan-free transgenic plants. So we will transfer plant genetic material by the use of the following methods. Next, DNA delivery methods available to produce plant transformations. We also used some modern techniques for the transformation of genetic material. Non-biological based transformation, direct method, and biological gene transfer is a indirect method. Direct method, DNA transfer in protoplast, chemical stimulated DNA uptake by protoplast, electroporation, liposuction, microinjection, sonication. Those are the DNA transfer in protoplast and also the direct method. Some other direct methods, DNA transfer in plant tissue, particle bombardment, also known as biolistics, silicon carbide, fiber mediated gene transfer, laser microbeam induced gene transfer, and biological gene transfer or indirect method. indirect method, agrobacterium mediated transformation. Primary two methods, co-cultivation with the explant tissue. In plant, transformation mediated by viral vector. So, in the direct method, after the isolation of DNA, we will transfer directly by this method. And indirect method, after the isolation, purification of the specific gene or specific DNA, then we will use this method, agrobacterium transformation. First, we will isolate the specific gene. It is cut by restriction enzyme and ligated by the ligase enzyme, and it is attached with a vector. Then the vector is transferred to the host and multiply a huge number of copies. Therefore, cultivate with the explant tissue, and in plantar transformation, and also transfer mediated by viral vector. We also transfer different viral vectors. Those are M13 phage, HB40, and others, viral mediated by the transformation of plant cell.